Hey, good evening. I want to start with a story that you will not hear on the news. And here's the story. Basically, what we've got is an amoeba in the ocean. But this one is a really big amoeba. This is an amoeba that is enormous, bigger than a curious diver who swims over to take a look. The amoeba stretches out its pseudopods. Pseudopods means false feet. So the amoeba stretches out its pseudopods and engulfs the diver within a food vacuole. That food vacuole, oh my goodness, winds up being fused with a lysosome, which is an organelle that's filled with hydrolytic enzymes. And then you know what happens? The poor diver gets dissolved. Okay, it's not real. It's something you'll never hear about on the news. But in today's show, we're going to talk about why that is. Why don't you see amoebas that are bigger than people? Here's another thing that I just couldn't resist showing, being that I grew up on Star Trek in the 1960s, the one with Captain Kirk, played by William Shatner, and Leonard Nimoy playing Mr. Spock. There was actually a show in which a gigantic amoeba was moving through space. That's a problematic idea in and of itself. But what was really problematic was the size of this amoeba. It was tens of thousands of miles in diameter. It was swallowing up planets. So the question is, could such an amoeba actually exist? Or really the question is, why aren't things like this existing in our universe? And so the question is, that's what we're going to talk about in the AP Bio Show. We're going to be talking about the size of cells. And actually, let's talk about what we're going to talk about tonight in the AP Bio Show, episode 6, which is the show that's designed to ensure your success in AP, IB, or any other kind of biology, like a freshman college biology class. So what we're going to talk about tonight are the issues related to the size of cells, and that's all about the surface area to volume ratio. In addition to that, we're going to talk about cellular compartmentalization, how eukaryotic cells are organized, and we'll talk about eukaryotic cell parts and their functions. Now, one thing that I would really appreciate from the folks in the audience is this. Can you hear me? Can you hear me at all? There's a little bit of drama that I had. And Fazlu, can you hear me at all? How's my audio sounding? Sound pretty good? So anyhow, let me know. Say you can hear me okay, and then I'll feel confident and I'll be able to go on with the show. So Fazlu, we're all depending on you to just post a comment saying, Mr. W, you sound fine. Go ahead. Let's do this thing. And fantastic, Fazlu. Okay, feel much more confident now. Okay, so in addition, tonight, talking about the size of cells, the issue of cellular compartmentalization, and the parts of cells, we're going to have a song of the week. There's going to be a weekly quiz. As always, there's going to be time for your questions, and there's going to be tips for learning. Listen to this. biology. That's my class's battle cry. Okay, listen, I've been doing that for 25 years. You can ask any of my students at Berkeley High School. So long before there was Chappell Rowan, who was doing HOTTOGO, I didn't exactly do it in exactly the same way, but I did it. I did BIOLOGY. That's our class's battle cry. And that's what we've got going on tonight. We've got some BIOLOGY, learning a little bit about me for those of you who might be joining me for the first time. My name is Glenn Wolkenfeld. I'm also known as Mr. W. I'm a retired AP Bio teacher, but not really retired because I'm still teaching. And you know who I'm teaching now? I am teaching you. And I am really, really psyched to be here with you, learning, teaching some biology tonight. My mission is to help you succeed in AP, IB, or college biology. So the more people who are here, the better questions we have. Take out your phones right now, text a friend, tell them to hop on to the AP Bio Show because I'm really happy to be here with you. One way that I'm really going to help you succeed is through a website that I created. It's called learn-biology.com. I also have an app that's called Biomania. I'm going to tell you more about both of those later on in the show. Here, it's time. Let's go into the lesson of the week. We're going to start with cells, a basic introduction. And I want to emphasize that what I'm going to do is I'm going to get into a groove, I'm going to do some teaching, but I'm going to stop very frequently and I'll pause and I'll say, folks, any questions? And that's really what this is all about. The beauty of YouTube is that it's a live show, and I am looking forward more than anything to answering your questions. So, ready? Strap your seatbelts on. Let's go. Let's talk about the structure and fundamental parts of cells. So cells, as you know, you're all taking a biology class, I'm assuming, are the basic units of life. They're the basic units of structure and function in living things. And this is a very diagrammatic representation of how they're made up. So all cells have a membrane on the outside. The membrane distinguishes the inside of the cell from the outside. And the inside couldn't be more different from the outside because cells are super organized entities. And the membrane separates that super high level of organization from the relatively high level of entropy, disorganization outside the cell. All cells have some genetic information, and that's in the form of DNA. And there are systems that cells have for maintenance and replication. And one of the most important of those systems is a system by which messenger RNA brings information to ribosomes and makes proteins. That's the central dogma of molecular biology. It's super important. And those proteins, most importantly, are enzymes that control the cell's metabolisms, that enable the cells to take in what it needs from the outside, to process it, to reproduce, to repair itself, and then ultimately to release waste that exit the cell, releasing entropy into the outer world. A big concept is the difference between prokaryotic and eukaryotic cells. We'll start with prokaryotic cells, only out of respect for the fact that they were here first. Prokaryotic cells have been around since the dawn of life, and that takes us back about 3.8 billion years ago. Prokaryotic cells are small, relatively simple, though they're extraordinarily complex. They have no nucleus. That's kind of what prokaryote means. Their chromosome is circularized. In other words, it's a DNA where the end and the beginning join up to create one continuous loop. They contain extra chromosomal pieces of DNA called plasmids, and they're found in two of life's three domains, the archaea and the bacteria. Eukaryotic cells, by contrast, are larger, much more complex, and this diagram is completely out of scale. The eukaryotic cell would dwarf the prokaryotic cell. Eukaryotic cells have a nucleus. They have multiple linear chromosomes. In other words, there's one end and another end. And if you were on the show a couple of weeks ago, we talked about the Hayflick limit. That's part of the explanation for why we don't live forever. Their DNA is associated with proteins, and they have mitochondria. Mitochondria are really the thing that define eukaryotic cells. In other words, eukaryotic cells only exist because of mitochondria, though they, of course, are distinguished by their nucleus and other features. And eukaryotic cells have many organelles, specialized parts that are surrounded by a membrane. Here's an expert tip for you, just to preview some stuff that we'll be talking about later on in the year. Prokaryote is actually a bad evolutionary category. The prokaryotes aren't a unified group. Prokaryotes don't derive from a common ancestor in the way that, say, all the vertebrates do or all the mammals. Those are all good, solid evolutionary groupings. But the basic idea is that bacteria are one kind of prokaryote, and on a molecular level, they're quite different from the other prokaryotic group, archaea. That molecular level involves looking at things like the sequence of RNA bases that make up ribosomes and things like that. It's fairly deep biology, but bacteria are no more closely related to archaea than they are to eukarya, in fact. And What we'll see later is that the eukaryote, the third domain, actually came about through, and this is unbelievable, a fusion of these two different domains, bacteria and archaea. We'll talk about that later. Let's move on to the issue of cell size. We started this show talking about why there aren't gigantic cells, like, for example, an oceanic amoeba that's capable of swallowing up a scuba diver. Well, cells are small, and the reason why cells are small, and I put a range of size there, it's from about 0.1 micrometer to about 100 micrometers. And 100 micrometers, just so that you can have some sense of this, if you can imagine a millimeter, 100 micrometers is 1/10 of a millimeter. So that's pretty small. I mean, a millimeter, clearly visible, 1/10 of a millimeter, just visible. And it all comes down to diffusion. And the basic idea is that cells need to have enough membrane surface area to allow for diffusion of substances in and out. And that issue of having sufficient membrane surface area, that's all about cell size. Let's look at how this works. If you measure the relationship between surface area and volume in a cell, then what you find, not only for cells, but for any object, is that as an object gets larger, its amount of surface area relative to its volume. Okay, we're talking about a relationship between surface area and volume. So the amount of surface area relative to the volume decreases. And let's look at a little math. We have two cells here. One is 1 micrometer and the other is 10 micrometers. So let's look at surface area and volume in these two cells. These cells are cubic, just to make the math really easy. So for a cube that is 1 unit of anything in size, then the surface area is 6 times the side squared. So 1 times 1 times 6. So there'd be 6 units of surface area. The volume is 1 times 1 times 1, 1 cubed. So the ratio of surface area to volume is 6 to 1. 6 units of surface for every 1 unit of volume. Now, if we take a cell that's 10 times bigger, 10 micrometers on a side, but really it could be anything, then what we have is 10 times 10 times 6, 600. Wow, you might be thinking, that's a lot more surface area. But it's not really a lot more surface area, because look at the volume. The volume's up to 1,000, 10 times 10 times 10. And so the surface area to volume ratio is 0.621. It's only 1/10 of that of the smaller cells. And we're going to go more into depth about this, but I just want to take a moment and acknowledge Ram's question. And we're going to get to your question as soon as I finish this first section. Then I'll open it up for questions and we can talk about anything. So including your question about FRQs. So here's what I just said. The larger cell surface area to volume ratio is 1/10 of that of the smaller cell. Now, a consequence of that is that large cells can't efficiently use diffusion to get the nutrients they need in and to get wastes out. Large cells really can't effectively use diffusion for exchange of materials. If you're in a typical AP Bio class, there's a lab that you'll do where you'll make cubes that are made of agar. Agar is a seaweed extract. It's the thing that you grow bacteria on in a petri dish. And these are cubes that have an acid base indicator on them. And what you do is your teacher will make up the cubes for you. Then you get the cubes, you cut them into various sizes, and then you soak them in vinegar. And in this acid base indicator, what will happen is that if they're basic, they'll be this beautiful magenta color. And if they're acidic, which is what vinegar is, then they'll turn white. So here we've got 1, 2, 3, 4 cubes. And if I'm remembering their size, it's something like 3 centimeters on a side, 2 centimeters on a side, 1 centimeter on a side, and then 0.5. You let them sit in the vinegar for the same amount of time. The vinegar is diffusing into the cell. After X number of minutes, let's say it's 5 minutes, the smallest cube has all of its volume completely reached by the vinegar. It's completely reached by diffusion. whereas the largest cube only has 19% of its volume reached by diffusion. If this were a cell, then, well, this outer portion would be getting enough oxygen. It would be able to get rid of waste, but all of this inner material would be choking on waste and would be starving. That's why cells are small. Cells are small in order to give them enough surface area to volume so that they can exchange things by diffusion. That's why huge cells are impossible. Their insides would starve, they would choke on their wastes. So how did life become large? Life became large not by having bigger cells, but by multicellularity and a host of adaptations that increased internal surface area to allow for the diffusion of materials in and out. Here we have two multicellular organisms. This is a beautiful kind of algae. It's called a volvox, and this is a kind of worm. It's called C. elegans, and it's interesting for a couple of reasons. It's only like 1 millimeter long, but it's incredibly studied in terms of developmental biology. And this whole organism is only about 900 cells, and biologists have been able to map out the cellular development of this organism. It's quite extraordinary. So let's look at some adaptations for increasing surface area. One is, let's just be flat. This is an organism that's called a planarian. It's extraordinarily flat. Flat things have a lot of surface area relative to their volume. Think of a piece of paper. It's all surface. There's very little volume. So planarians don't have a circulatory system. They don't have a heart. They don't have blood. They have a digestive system, but the food just diffuses to their cells. They don't have a circulatory system. They don't really have a respiratory system. It's all about diffusion. Why do elephants have huge ears? Those ears are radiators. What they do is they provide a lot of surface area, allowing for the diffusion of heat. Why? Because an elephant's a big animal, and it can't easily, through its body surface, exchange heat with its environment. So it has evolved these huge ears. These are some dissected out gills. Thin flaps of tissue have lots of surface area. That's how fish absorb oxygen that they can bring to their blood. This is the inner lining of the mitochondrial membrane. It's not a lining, it's the inner mitochondrial membrane. And what is it for? It's because there are all these embedded enzymes that create ATP. To have enough room for enough of those enzymes so the mitochondria can create lots of ATP, that inner membrane is highly folded. This is the inner surface of your intestine. Highly folded, folds within folds, so that it can absorb food into your bloodstream, which will then circulate it to your body. So, increased surface area for diffusion of molecules of heat, increased working surface for membrane-embedded enzymes, and it's through thin sheets of tissue or highly folded surfaces. So here's another example, and then we'll end this topic and we will open it up to people's questions. In terms of surface area, there are no small, like mouse-sized marine mammals. Think about all the mammals that live in the water. The smallest one is the otter. And some of the biggest, in fact, the biggest animals that have ever lived are marine mammals. Whales, like the blue whale. Why are marine mammals big? Why are they never small? They're never small because mammals are warm-blooded, and you lose heat through the outer surface of your body. The smaller you are when you're like a mouse, then you have a lot of surface area relative to your body. So the heat just diffuses out of the body. That's disastrous because you would become hypothermic very quick in cold water. And it doesn't have to be really cold water. It can be water that feels warm, like 70 degrees. But over time, your body temperature will wind up equilibrating, or you'll just wind up using up all your internal energy trying to maintain heat. So increased size decreases your surface area to volume ratio. Whales are able to withstand the cold of the ocean just because they're big. They have relatively little surface area relative to their volume. And again, it's a relative thing. Of course, a whale has a huge amount of surface area, but relative to its even huger volume, it doesn't have much. Biology, so cool. This concept, surface area to volume, is one of the most powerful concepts in biology. It explains so much. And now It's the time for questions. So you got a question about surface area, volume, or anything else, put it in the comment thread right now, and I will do my best to answer. I am so happy that those of you who are watching me right now. And of course, you can put your question in the comment thread, and I'll get to it in a little bit too if I don't get to it right now. So, Ram, if you're still there, you can put your question in, and if not, put it in, and I'll get to it at the end of the next section. So, any questions coming in? Anybody curious about anything about surface area and volume? Wondering about that? Okay, not seeing anything. Maybe Ram is furiously typing. All right. Listen, Ram, just get the question in, and when I get to the end of the next section, we'll talk about it. Okay. We're going to move on to the next part of this 3-part lesson before we get to some of our special goodies at the end, our song of the week and our quiz. We're going to talk about cellular compartmentalization and the endomembrane system, which is really about how eukaryotic cells are organized and how that organization evolved. So, first of all, let's talk about what this is, compartmentalization. Take the word apart and it'll tell you what it means. Compartmentalization is internal division into sections. When you go into a plane, there's a luggage compartment. When you go into a bus, there's a luggage compartment underneath the seats. So those are compartments. The advantages of compartmentalization is that it allows cells to have regions with internal chemistry that's distinct from the cytoplasm. So for example, this organelle over here at number 7, that's a lysosome. It's filled with hydrolytic enzymes. We talked about hydrolysis in a previous part of the show. I'm actually going to change my view over here, make myself a little bit smaller. I'm going to go like this. Okay, probably easier for you to see right now. So this is a lysosome over here. And what the lysosome does is it's filled with hydrolytic enzyme. It's kind of like an internal digestive organ. Trying to get that laser pointer. Fantastic. And you don't want those hydrolytic enzymes released into the cytoplasm because they would break down the cytoplasm. So how does a cell handle that? Through compartmentalization. Another thing that compartmentalization does is it increases surface area for membrane-bound enzymes. So like, for example, the smooth ER has a lot of enzymes. Those enzymes can only work in the context of being embedded in a membrane. Well, let's build lots of membrane that produces lots of surface area. And ribosomes in the rough ER are another example. Those are protein factories that only work when they're embedded in a membrane, because that wasn't really true. Those are protein factories, and they can work either in the cytoplasm or embedded in the membrane in order to produce the kinds of proteins that will be used for export or for positioning within an organelle. They have to be within a membrane, and that's why there's a rough ER. Let's talk about compartmentalization in prokaryotic and eukaryotic cells. In prokaryotic cells, there's hardly any compartmentalization. There's minimal compartmentalization, though there's some. So like, for example, when we study about photosynthesis, we'll see that there are these sacs that are called thylakoids, and those are compartments that enable the light reactions of photosynthesis to happen. I'm showing them in a chloroplast, but they're also in free-living cyanobacteria. Those are photosynthetic bacteria. And there's also eukaryotic cells, which are highly compartmentalized. Many internal membranes that divide the cell into regions with distinct structures, chemistry, and function. And examples of those compartments are lysosomes, the endoplasmic reticulum, the Golgi complex, and vacuoles. Many eukaryotic compartments are part of what's called the endomembrane system. And that's a dynamic system of internal membranes and compartments. So here we have the nuclear membrane, which is contiguous with the rough ER, which is contiguous with the smooth ER. The smooth ER buds off vesicles, which go to the Golgi. The Golgi buds off vesicles that go to lysosomes and go to the membrane. Phospholipids are moving between these various parts. So I'm showing a static picture, but you have to imagine a lot of dynamism of parts moving from one part to another. Membrane and materials flow from one compartment to the next. How did compartmentalization come about? We're explaining the origin of mitochondria, cellular compartmentalization, and chloroplasts. It's really the origin of the eukaryotes, which means, hey, this is what's so cool about biology. It's about the origin of you and me, because we're eukaryotes. This is how our kind of cell came into the universe 1.8 billion years ago. So the basic idea is that this arose as a kind of mutualistic endosymbiosis. Mutualism is when two beings help one another out in a way so that both benefit. If you are studying biology and you have a study partner and you both help each other, that's a mutualistic relationship, as opposed to, for example, if you cheat off somebody else. That's a parasitic relationship because, well, it's not necessarily parasitic. If you harm someone else as you take from them, that's a parasitic relationship. So mutualism, it's a win-win. Endosymbiosis is living together. Endosymbiosis is when one thing lives inside another. So back to parasitism. If you get infected by a parasite, it can be a bacterial parasite or a worm or something like that. That's endosymbiosis, but it's negative endosymbiosis. This is positive mutualistic endosymbiosis. So what happened? In the scenario that I think makes the most sense, there's an archaeal cell that took up a bacterial cell. The two cells were living in close association, probably already exchanging materials, but at one point the bacterial cell phenomenally went inside the archaeal cell, and the archaeal cell did not digest that cell. It either allowed the cell to continue to live inside of it, the bacterial cell, or the bacterial cell had some mechanism that kept itself from being digested. But in any case, over time, that bacterial cell evolved into a mitochondrion. And secretion of vesicles from that bacterial cell that was to become a mitochondrion led to the nuclear membrane, the endoplasmic reticulum. Later, a second endosymbiotic event led to chloroplasts being taken up by an early eukaryotic cell, and that led to the origins of the algae and the plants. So endosymbiosis is how the eukaryotes came together. This is a story that was popularized by a scientist named Lynn Margulis, who I totally encourage you to learn more about. A really important, groundbreaking scientist. So just to take another look at the three domains, what I want to point out is that bacteria were taken up by archaea, and that led to the eukaryotes. Our domain, all of us eukaryotes, the plants, the fungi, the animals, they're all combination of these two other domains. And then later, cyanobacteria, that's a free-living bacterial photosynthetic organism, was taken up by an early eukaryote, and that led to the plants and the algae. It's quite an extraordinary story. When you think about it, that means that you are kind of a big colony of bacteria, because that's what mitochondria are. It's pretty phenomenal. What's the evidence for this astounding idea? Like bacteria, both mitochondria and chloroplasts have their own DNA. Why would they have their own DNA? Because they were once independent cells. They replicate themselves through binary fission. We don't replicate our mitochondria, they replicate themselves. They have their own ribosomes, and they produce some of their own proteins. And those ribosomes are like bacterial ribosomes in terms of their RNA sequence and their structure. Also, both mitochondria and chloroplasts have two membranes, and that is a vestige of the fact that they were once engulfed by another cell that surrounded them with a membrane. Biology blows my mind. So amazing. I'm so glad to be here learning it with you. All right, another opportunity where we're going to slow down and see if we've got any questions or any comments. Anybody got any questions? Anybody got any comments? Because I'm more than happy to answer them. In fact, I'd say that's the most important thing that we're doing here tonight. Me being available for you to help you learn biology and help you succeed. So if you got a question, put it in the chat. If you got a comment, if you just want to say, hey, Mr. W, doing good. Keep going. Want to hear that too. So thank you so much. All right, let's go on because I'm not seeing anything come into the chat. Okay. By the way, there's a lot of intense stuff that we've just talked about, stuff that ranges over 4 billion years of biological evolution, and stuff that involves some math and some concepts. And so if you want to learn more, I've got tutorials waiting for you at learn-biology.com. It's an AP Bio website that's been approved by the College Board for use as a textbook. And do the tutorials. You won't believe how much you'll learn. And you can try them out with a free trial. All right, the last part of tonight's lesson, eukaryotic cell parts and their functions. So let's talk about the nucleus. The nucleus's function is it stores and protects genetic information. Genetic information, the DNA that we inherited from our parents. The DNA is wrapped around proteins. Those proteins are called histones. You'll learn all about that in Unit 6, and they form structures that are called chromosomes. Here's a chromosome over here. But most of the time during the cell's life, the DNA is spread out in a diffuse form that's called chromatin. There's a dark spot in the middle of a nucleus. You can see it when you look at the cells with a microscope, which is something that I hope you do in your biology lab. You see a dark spot. It's called a nucleolus. Here, it's over here. And what it is a ribosome factory. It assembles ribosomes. The nuclear membrane separates chromosomes from the cytoplasm, and nuclear pores are found throughout the nuclear membrane, and they allow molecules to enter and leave. So, for example, we were talking before about how messenger RNA is sent from the DNA to ribosomes. That messenger RNA leaves the nucleus through a nuclear pore. But there's other molecules that come in that act as what are called transcription factors that tell the nucleus what mRNA it should be producing. So there's communication that goes back and forth. What about ribosomes? Those are particles that are composed of RNA and protein. That RNA, because of where it is called ribosomal RNA, and they consist of a large subunit and a small subunit. The ribosome's function, it reads a genetic message that's encoded in a sequence of mRNA. So here, the ribosome is reading mRNA, and it translates that message into a sequence of amino acids that make up the primary structure of a protein. In a previous episode of the AP Biology show, I discussed how there are 4 levels to proteins. You can go back and watch that episode again if you need to. The details of translation and protein synthesis are mind-blowingly cool, and we will talk about that when we get to Unit 6 much later in the year. Where in a eukaryotic cell can ribosomes be found? I tripped over this earlier in the show, but ribosomes can be free or bound. Free ribosomes, like those shown at 3, are floating freely in the cytoplasm, whereas bound ribosomes at 4 are connected to the membrane of the rough ER, the endoplasmic reticulum. The key idea, and again, this is another thing that you can learn about on learn-biology.com, is all ribosomes start out as free. And through a process called protein targeting or signal translocation, they migrate to the ER to become bound. There's a tutorial about that on learn-biology.com. What about the structure and function of the mitochondria? Well, the function is converting food energy into ATP. ATP, as we discussed a little bit earlier in the course, but this is really a topic that we'll hit on in Unit 3. This is the molecule that enables your cells to get work done. Every time you contract a muscle, every time you think a thought, well, for a muscle, it's direct. For the thought, it's a little bit indirect. But all of those things wouldn't be possible without ATP, which enables cells to do work. The key structures in the mitochondria, going from the inside out, there's a matrix, which is essentially the cytoplasm of the mitochondria. Why does it have cytoplasm? Because it was once an independent cell. Those consist of enzymes for the Krebs cycle, a very important reaction that we'll learn about in Unit 3. There is a chromosome. Mitochondria have their own DNA that's shown at number 3. They also have ribosomes, which aren't shown in this diagram. Those are both evidence of the fact that mitochondria were once free-living bacterial cells. The inner membrane, which is shown at 2 over here, it's highly folded, and that's an adaptation for increasing surface area, which enables the mitochondrion to produce more ATP because it takes a lot of embedded enzymes, protein pumps, things like that to produce ATP. And that is what I just said. There's an intermembrane space, which is also important in the process. It enables the mitochondrion to trap protons, and the diffusion of protons is later used to synthesize ATP. The outer membrane at 1 is a vestige of endosymbiosis, and that's what mitochondria are all about. The endoplasmic reticulum, it's an interconnected series of channels between the nuclear membrane and the Golgi body, and it's got 2 forms, rough and smooth. The rough ER, it's studded with ribosomes. So all these little dots over here, those are all ribosomes, and it synthesizes proteins that can get included in lysosomes, other organelles, into the membrane itself for export by the cell. And what I have over here is a picture that's really extraordinary. Every time that you fight off an infection, you have immune system cells that are called B cells. When those B cells get stimulated to fight off an infection, what happens is they build up a lot of rough endoplasmic reticulum. Why? Because they're producing tons of proteins that get secreted into the blood that fight off whatever pathogen has invaded you. What about the smooth ER? It's on the outer side of the ER network. So here's the rough ER, here's the smooth ER. It lacks ribosomes, but it still has many embedded enzymes, and the functions vary by tissue. So it can be synthesis of lipids, it can be converting toxins into soluble forms that can be excreted from the body, and it can be carbohydrate breakdown and synthesis. And I'm seeing a question from Ram that is just filling me with joy. So I really want to encourage you guys to ask questions. Does the Golgi body or the rough ER give newly made proteins their tertiary or quaternary structure? I would think about it this way. The rough ER has these ribosomes that are producing proteins. Those proteins that are initially emerging are probably in a tertiary form, and then they would probably be combined later. Then that can happen in the cytoplasm, or it might happen in the Golgi. The Golgi has enzymes that are trimming off pieces of proteins or adding little pieces. The Golgi is frequently thought of as being like a packaging and sorting center. So the Golgi will package proteins in vesicles. Those vesicles will have little molecular tags, and those tags will tell the proteins where to go. Are they destined for export? Are they destined for the membrane? And so on and so forth. So I don't think it's really clear about whether they would leave one area in tertiary structure and then become quaternary somewhere else. But basically, as protein synthesis ends at the ribosome, the tertiary structure is already emerging. So that would be happening in the rough ER. It would also be happening in the cytoplasm. And it could be in the Golgi that these polypeptide chains are put together. But I need to learn more about that, Ram. So I'm very grateful to you for asking that question. And I'm not 1,000, or I guess you can't really be more than 100% sure. But thank you very much for that question. It's really the best I can do on that one. The Golgi complex, what we were just talking about. It's a series of flattened membrane-bound sacs. It receives vesicles from the rough and smooth ER and chemically modifies the contents, and those are usually proteins. And it packages modified proteins into vesicles that are sent to organelles to the cell membrane or exported from the cell. It's also known as the Golgi body or the Golgi apparatus. What about the structure and function of lysosomes? First of all, you're never going to have to identify a lysosome on the AP Bio exam. And that is because lysosomes are simply vesicles that are filled with enzymes. So they're clearly identified over here, but you can't really identify them, and don't worry about them, but do know their function. And their function is to contain hydrolytic enzymes. They're found only in animal cells. It's another important thing to know. And they carry out intracellular digestion. So I wasn't able to get this photo to show to you, but there is a photo of like a mitochondrion that's being engulfed by a vesicle and then fused with a lysosome. And that's how a mitochondrion, when it's worn out, will wind up getting digested by the cell. And that's what I just said. They recycle worn out organelles, damaged organelles, and they play a key role in what's called apoptosis, which is programmed cell death. I'm getting a question about knowing about chromosomes, and do you have to know about them in depth? No, I would just know at this point in a Unit 2 test that chromosomes are how DNA is packaged up. You'll learn later about the details of that packaging because that's important in relationship to gene expression, but that's mostly a topic that's in Unit 6. Now, I do want to say that every teacher is going to teach this material in a different way, but when I taught AP Bio, I was mostly getting my students to understand the basic structure and function of these various parts. Unit 2 is kind of like an overview, and the details happen later. And Priyanthan, thank you very much for that question. Great question. What about the cytoskeleton? It's a dynamic network of protein fibers. It enables cells to move materials and organelles and enables cells to move their membranes. So even though amoebas can't swallow up divers, they can swallow up other things like bacteria. So can your white blood cells. Your white blood cells roam your body looking for infecting cells, and they'll also stretch their membrane around an invading cell and engulf it in this process that we'll learn about later in Unit 2 that's called endocytosis. Centrosomes, centrioles. The centrosome is the organelle. It has two centrioles. And those centrioles, what they do is they create a spindle of fibers that separate chromosomes during mitosis and meiosis. And those are Unit 4 and Unit 5 topics. And Devika, thank you so much for asking me this question. I am trying to do a live show every Wednesday night at 6:30 p.m. Pacific time. All right, so you can count on me here, but you know, if you can't make the live show, you can always watch it on replay. What about the central vacuole? It's only found in plant cells. Its functions are water storage, storing and releasing needed molecules, and sequestering waste products. Evolutionary origin of chloroplasts, they were once free living cells, just like mitochondria, and they took up residence within an early eukaryotic cell. Their function is creating carbohydrates through photosynthesis. The details of how chloroplasts work and the details of photosynthesis, which is like the coolest process imaginable, that will happen in Unit 3. Plant cell walls, chemical composition. They're made of cellulose, that's a polysaccharide. We talked about that in Unit 1. Their major function is they act as a pressure vessel that prevents overexpansion in response to inward water flow. It's called osmotic pressure. And, you know, paper, wood, cotton, those are all things that are made of cellulose. It's one of the most important substances in our civilization. Any other questions or comments? All right, well, listen, I was going to do a song of the week, but I think that what I'm going to do is move into a very popular part of the show because the song that I was going to do was the Cell song. I had some technical problems that are keeping me from doing it live. I'll save that for later once I figure out how to do that. And I'm seeing a very, very sweet question. I've already got two kids who I love, but listen, I will be your biology teacher. That's what I will do. So thank you very, very much for that question. Just want to emphasize that, like, if you felt like, damn, he was going really fast as he covered this material, I've got tutorials about all this stuff on learn-biology.com. Will essential vacuole, and I've got a question from Rom, will essential vacuole being filled completely lead to increased water potential? I am going to teach about water potential next week. I've got a tutorial about that on learn-biology.com, and I think you've got the cause and effect a little bit messed up. So if the water potential outside of a cell is higher than the water potential inside the cell, then water will flow by osmosis. So higher water potential over here, lower water potential over here. This is pure water. You've just watered your lawn, right? So here's water, here's cytoplasm. The water potential is going to be higher over here than in here. Water will flow into the cell from higher water potential to lower water potential, and it'll fill up the vacuole. So that's the causality. Water's flowing from higher to lower water potential. And where does that water go? It goes into the cell. So that's the basic idea. And Ram, since we're right now at a good moment about question, what's your question about the FRQs? So go ahead, type it in. And while you type it in, I am going to set up not our song of the week, but our weekly quiz. So I'm going to click over here. You know what I'm going to do? I'm going to do this. You can't see that. All right, here we are. Go here. Excuse me. Okay, so Ram, never saw that question about the FRQ, but maybe that was a question about the FRQ. So I want to tell everybody what I'm doing. So right now I've gone out to learn-biology.com. That's my AP Bio Learning website. And now it's time for our weekly quiz. So what works best in this format is multiple choice quiz. I'm going to ask you a couple of questions, and what you're going to do is you are going to type in the letter of the answer. So I just chose multiple choice quiz 1 and got to log in. Fantastic, logging in. Okay, so we talked about cell parts and we talked about cell size, and that's really, and we did talk about compartmentalization and the origins of cell compartmentalization. So that's a lot of questions. We're not going to do that many. And and Ram is asking me a very general question about FRQs. What are some common mistakes that you see your students make that make it hard to give them a point credit for the question? So the mantra of FRQs is claim, evidence, and reasoning. So I think what I find is that when students don't give evidence to back up an assertion, that's often where they lose points on an FRQ. And of course, there's the most basic thing, which is like, you don't read the question. But one thing that I want to emphasize, I'm just going to show you this, is that I've got all of these practice FRQs on learn-biology.com. They're very short because, you know, I'm giving them online and they're not really like the College Board FRQs in terms of their length, but they will really help you in terms of getting good practice at developing specific FRQ answers. But let's do a couple of multiple choice questions. Okay, so everybody ready? Okay, first question. Okay. This is a tough question. This question would happen better later on in the year, but I think you can do it. Okay, so, and this is a very AP-like question. The diagram below illustrates a process related to glucose homeostasis in liver cells. And for those of you in the listening audience, we've got a membrane, there's an insulin receptor, there's a glucose transporter molecule, and then there's a bunch of molecules that are part of a cascade, and there's a vesicle that's budding off from something within the cell. The structure that the GLUT4 vesicle, that's a vesicle that has a bunch of glucose transporters, buds off from, is most likely what? And here are your choices. The smooth endoplasmic reticulum, that's 1. The Golgi complex, that's 2. The rough endoplasmic reticulum, that's 3. And 4 is a lysosome. You folks who have been watching know enough to answer this question. And I've seen one answer come in from Priyanthan. Who else is going to try and answer this question? Anybody else? Got a bunch of people watching, so I'd love to see more answers come in. Anybody else? Okay, let's go with, I hope it's okay to call you Priya. Answer shows, oh, we got a 4. Let's go with Priya's answer. The Golgi complex, yes. A typical pathway for the creation of membrane-embedded protein like the GLUT4 protein is shown below. The vesicle that brings a protein like GLUT4 to the membrane would be represented by 6. The vesicle would bud off from the Golgi, which is shown at 5. So that's the kind of questions and feedback that you get at learn-biology.com. Let's do another question. Which letter in the diagram represents the rough endoplasmic reticulum? We've got a cell that's shown with its membrane. We've got a structure that has a dark spot in the middle. Outside of it, there's a series of channels with dots, and then there's a series of flattened sacs, and then there's a membrane. So we've got A, B, C, or D. What do we have here? We have A, B, C, or D. And I see one answer that's come in from Ram. Any other answers? What represents the endoplasmic reticulum? And let's go with A. Nice job. A represents the rough ER. You folks are doing fantastic. Next question. KDEL is a short polypeptide sequence consisting of 4 amino acids: lysine, aspartic acid, glutamic acid, and leucine. As certain proteins are being synthesized and or modified, KDEL binds with a specific receptor. This receptor, in turn, prevents the protein from being secreted in a vesicle for further modification. Based on this description, the KDEL receptor is most likely found in the plasma membrane, the cytoplasm, cytosol, the endoplasmic reticulum, or the mitochondria. So what do we have here? What do you think? Let's call this A or 1, B. Let's go A, B, C, or D. Where's KDEL? Can you see in a couple of choices? All right. Seems like A is the majority. I'm going to click on A. No. Take a look at the diagram below. The only way that a protein is going to get to the membrane would be through a vesicle, such as the one at 6 or 4 below. Using the diagram, see if you can find a location where proteins are synthesized or modified before they would be incorporated into a vesicle. That's where you might find the KDEL receptor. So what I want you to notice is what we do on learn-biology.com. If you get it wrong, you get a hint. And then you get to try that question again. These questions are like a stack of cards. So let's do one more question. Okay, in a marine food chain that ends with a humpback whale, phytoplankton are the first link. Phytoplankton, that means plants that are tiny cells that float around in the water. Which of the following statements apply to both the mitochondria of humpback whales and the chloroplasts of phytoplankton? 1, contains its own DNA. 2, makes ATP by chemiosmosis. 3, temporarily stores energy in reduced electron acceptors. Guys, this is a tough question. Tough question. This is really like an end of the year kind of question. So anybody want to give it a try? Okay, so the truth is that the answer is, I believe, 3. 1, 2, and 3. All of those statements are correct. Yes, they have their own DNA. They make ATP by a process called chemiosmosis. You'll learn about that later. They store energy in reduced electron carriers. Folks, we could go on, but we've already exceeded our hour. So I'm going to jump back to my slideshow. And let's go like this. And I just want to tell you about your plan for this week. If you're in AP Bio, then what I want to encourage you to do is, if you're not already in a class, I mean, if you're in a class that's using learn-biology.com, then just do that, or do whatever your teacher tells you. But what I want you to do is go ahead and do some of these tutorials on learn-biology.com, because what you'll do is you'll get to read, you'll get to practice, you'll get to reinforce these ideas. It's really going to help you learn this material. You will make huge gains in your learning. Learn-biology.com is going to give you the skills and confidence to ace your biology class and the AP Bio exam. It's 100% aligned with the College Board. It's got interactive tutorials. It's got multiple choice questions, click and drag diagrams, identify the part questions, free response questions. You get positive feedback for correct responses. You get hints and repetition for incorrect responses. There's a student learning guide for every tutorial that tells you exactly what to do, everything you need to crush it on the AP Bio exam. And if you're a teacher, well, if you're a student, you can monitor your own performance. And if you're a teacher, you can get a class account and you can monitor all those students' performance. You can do it for free for 2 weeks. You can try it out, and then it's $24.95 a month after that. It's the best deal in biology learning. Any final questions to end the night? It's been a real pleasure being here with you, teaching you my favorite subject, biology. I'll see you next week on YouTube, and I hope to see you also on learn-biology.com. Please subscribe and also watch the next video. Ram, thank you. It was really a pleasure teaching you tonight. Everybody who was here, this was awesome. I'm looking forward to seeing you again next week. Tell your friends, the more people who are here, the more fun we're going to have. Everybody, be well, stay safe. See ya.